Jack Feldman in the house. And then we'll just keep an eye if other members are logging in as well.
Apologize for being late. There was traffic, which is an LA thing, I know, but it's been absent since COVID and now it's reappeared.
Not a problem.
Good.
Glad
Here we are.
Glad to be here with you.
Okay, so this is the Q&A.
Yeah, so welcome back, Dr. Jack Feldman. It's a pleasure to be here with you, and thank you for taking the opportunity to do part 2 and continuation from our conversation a few weeks ago, where we delved into quite a robust chunk of time together within the research that you have been exploring over the years within the pre-Botzinger complex, the neurons involved in the inhalation, and the parafacial nucleus in relation to active exhalation, discussing breath in multidimensional as a behavior, its connection to emotion, some interesting nuances into stroke and its connection to emotion therein. That was a beautiful story around the patient with his smile, to then dive even further.
Locked-in syndrome.
The locked-in syndrome case
Right.
that you were discussing, in addition to talking about opioids and related to breath and the suppression of the inhalation. So in that sense, would love to open up and allow for further conversation and exploration of all things breath with you. And firstly, I was curious to start off with a question for myself, and then open up the conversation to our awesome group here. And so in 2018, Wim Hof participated in a study at Wayne State University in Michigan. And this was an interesting study. I found it to be unique in that for many years, everyone's talked about brown fat and how Wim has these abilities to create heat in his body, and how to, from the inside out, meet the cold and allow for attenuation of that response. And what I found interesting in that study is that within the fMRI, they noticed this oscillation between the insula and the PAG, the periaqueductal gray. So this was a new finding in that, and the study being called brain over body, that we have, in fact, a brain over the body, and we have this ability for top-down modulation of the pain response in relation to cold. So was curious for your take, if you've seen that study or delved into it specifically, or any relation into the periaqueductal gray as related to breathing and attenuation of pain response.
Well, the periaqueductal gray plays a role in the pain response. I mean, that's very clear. It's a little hard for me to extrapolate from observations on one individual to things that may apply to everyone. Wim Hof may be truly representative of all of us, or he may be quite a bit different, in the same way that Michael Jordan can take off from the free throw line and stuff the basketball, make it look easy. But if I tried it, I'd look like a fool. So, you know, there are people who have exceptional abilities that are just part of the normal continuum of humans, but they may be very far from the mean or the average. And so I totally believe that the data they got on Wim Hof was absolutely spot on, but it's very hard for me to subscribe to any generalities from that. He's certainly capable of doing it, just like some people can jump over a bar 7 feet high. But whether or not we all can do it, or to improve in some degree, it's not entirely clear. Now, that said, I think a lot of things that we do with breath is accessible to everyone, that everyone can get something out of it. How much they get out of it may be in part how much they put into it, And it also may be limits to how much they can get out of it. Wim Hof obviously has figured out a way to get an enormous amount out of it, and it's sort of breathtaking what he is capable of doing. It's just not clear to me the degree to which the fact that he can do it, the rest of us can do it easily. That said, he has his method, and I know that you're a big proponent of that. And I think people who follow it diligently will probably see big changes in all sorts of aspects of their health that are positive. I don't know if that answered your question, and I'm not trying to stonewall you. I just, it's just a little difficult for me to go from a one-off, even if it's Wim Hof, to what you and I can do, or what you and I may not be the norm either, but what the population in general can do.
Indeed. Thanks for that reflection. And for example, someone in Wim Hof's case who has been practicing techniques of this manner for 30 plus years, that it brings me back to really where we concluded with breathing being non-prescriptive, and through continuous practice there and improving the communication between brain and body from top down to bottom up, that's where you can start to see that skill development or ability to deploy, as he says, by thought, to be able to turn on these certain regions of the brain, perhaps, and using the breath to amplify that.
Well, the brain is highly connected, and sometimes you want one part of your brain to communicate very strongly to another part, and that pathway may be weak or even non-existent. And I certainly think that there's lots of data that if there certainly is a weak connection, there can be ways to strengthen that. If there's no connection, I'm not so sure, but if there's a weak connection, I think that through practice of a variety of sorts, that in an effort to try and strengthen that link, it can happen. And so I think that could very easily underlie a lot of things that we're talking about here.
Fascinating. Well, I'd love to open the floor up for the group to pose questions. If we roll out with, for whoever does have a question that they would like to ask Dr. Jack Feldman, if you want to raise your hand or to unmute yourself and take it away, please.
Too shy?
Go ahead, Bryce.
Hi, Dr. Feldman. So great to have you speak with us again. It was a treat to get to listen to you last time, and it's another privilege. So a thought came to mind for me regarding possible therapeutic treatment of locked-in syndrome, given how involuntary abnormal breaths can transpire when given proper emotional stimulus. I was reminded of how visual cues, virtual reality, has been used to help people recover movement when they were thought to be paralyzed.
Mm-hmm.
And as someone in the performing arts, I'm a musician and I've done acting. I was reminded of how it's the line between what's genuine emotion and what is strictly physiological response, top down, is pretty blurry. Like, we've practiced two different types of breaths. One is the prototypical alarm breath. And just when I tell myself, eyes go wide, shoulders go up, sharp inhalation through the mouth, automatically I feel emotion in my body.
Mm-hmm.
And similarly, when we do this prototypical predator exhale with tongue out, biting muscles activated, and exhalation, automatically I feel this aggression pop up in me.
Mm-hmm.
And I was wondering if you think I know it's a very hypothetical question, but I'm curious about it. If you think there is any possible therapeutic possibility of linking the emotionally prompted breath to sequentially less emotionally prompted breath to teach people how to reconnect those nerves, the neural pathways.
Okay, if we're talking specifically about locked-in syndrome, the idea is that it irreversibly damages the connections from the cortex, which is up here, to targets that are below that. And this is the pathway by which volitional commands get transmitted to the limbs, to the chest, and so on and so forth. As we discussed last time, the structures that are involved in the regulatory part of breathing are below that damage, so they're fine. So what you're proposing is that you could go down through this emotional pathway, but ultimately hook up with volitional movements. And I won't say it's not possible, but I don't know of anyone who's actually tried to do that experiment. I would harbor a certain degree of skepticism about the ability for one to do that, but until you actually get a patient who has locked-in syndrome or some other you could take this at any level. I mean, spinal cord injury often involves a damage to the pathways that are descending, and where you see a loss of movement is below that. So your idea could be used across the board, not just in locked-in syndrome, but in any spinal cord injury that's affecting the descending command, and see if you can activate it. Now, there is an instance with the diaphragm, actually, called the cross-phrenic phenomena. So the phrenic nerve is the nerve that innervates the diaphragm. It comes out of the lower cervical spinal cord and goes to the diaphragm. If there is damage to the signal coming from the brainstem seems to cross and go down each side. And so if you damage one side between the prevascular complex and the other structures in the brainstem and the phrenic nucleus on one side, the initial response is that one side of the diaphragm will be not respiratory. However, within a couple of days, the pathway on the other side grows over. You don't have to do anything to have this happen. It's been reproduced in all sorts of experimental animals, that the pathway may not normally exist, but when the inputs are absent on one side, the inputs from the other side essentially grow over. And suddenly you get a restoration of activity on both sides of the diaphragm.
That's interesting, because I've heard from...
That's not generalizable. That seems to be very specific to the diaphragm.
I know Sam has worked with someone who has one half of their diaphragm paralyzed. And I wonder why that transfer of neural firing hasn't taken place.
Well, it depends why their half of the diaphragm is not working. So this so-called cross-phrenic phenomena occurs when you damage the axons above the cervical level of the phrenic motor neurons. So there's C5, C6. You have damage at C2, and it damages those axons. You see that. But you could also lose the hemidiaphragm if you damage the phrenic motor neurons on that side or the phrenic nerve on that side. So, Sam, if he's not violating HIPAA, might be able to tell us what the origin of the semi-diaphragm paralysis is.
Yeah, this is an interesting case study. I believe it was stress induced with then a virus that made its way in parabrachial to then inhibit and shut down the left phrenic nerve, left hemidiaphragm.
So if it's at the level of the nerve, then it doesn't matter what you're able to do in the spinal cord because it can't get out. It has to go, the signal has to pass through the nerve. So if the nerve is compromised in any way, you won't see any activity going out to the muscle.
And what's been interesting with this specific case is through every few months going in for testing and seeing with a sniff test that the left hemidiaphragm goes up as the right hemidiaphragm contracts with their inhalation. But over time, with some more mechanical practice around breathing movement, there has been slight improvement developing into the left hemidiaphragm. And in that case, that the left phrenic nerve is starting to fire and come back online.
I also could be that some of the axons in the active diaphragm have grown over to the other side. So I don't know what the status of that is, whether or not those axons will go across the midline. So there's a variety of possibilities. I don't know how easy it would be to do the experiment to determine what the case is.
That's interesting. Something to delve further into. Awesome. Thanks, Bryce.
Now, did I talk to you last time about episodic hypoxia?
No.
I mean, if there's no questions, maybe I could talk to you a little bit about episodic. Oh, do you have a question?
I do actually. I'm just curious about, like, you know, as a breath facilitator coach, around like the quantification of a breath practice. You know, I know like HRV being something that people pay a lot of attention to. Just wondering your thoughts on any other ways to actually quantify for someone. You know, I kind of have this idea around our collective. You know, we teach these daily breath practices, and figuring out a way for people to actually quantify how that is affecting them. I mean, we can, you know, do a state check on a felt sense in our body, you know, probably track a few other things, but just kind of curious around, you know, any tools or thoughts about how someone might actually be able to put some deeper, you know, numbers or quantification behind a daily practice sort of thing.
Okay, so I'm sort of spitballing here.
Yeah, great. Love it.
You know, one thing is blood gases. So, you know, we all presume that our oxygen levels are about 100 millimeters of mercury and the CO2s are about 40, but in fact, it could vary across people. There's a group of individuals who have clinical anxiety who tend to hyperventilate, and their CO2 levels are below normal. They're hypocapnic. There's a therapeutic regimen where these individuals are taught to breathe slower, and they restore their CO2 levels. And the claim is made that when they restore their CO2 levels to be normal, their anxiety gets reduced. So one is blood gases and pH is one measure. Now, pulmonary physiologists measure a whole bunch of things related to the, you know, things like lung capacity, forced expiratory volume, you know, is how hard can you exhale, how hard you can inhale. So there are all these parameters of lung function and muscle function, which a pulmonary physiologist or a pulmonologist, an MD, can measure, and those probably will be impacted by a breath practice. As far as breathing pattern goes, you know, you can make measurements, but I don't know where the data lies in terms of showing that there are changes and that these changes are associated with a improved outcome for whatever, you know, non-respiratory variables you're measuring, like emotions, stress, anxiety, panic, fear, and whatnot. I don't know.
Right.
I mean, I don't know if that, does that sort of address your question?
Yeah. I mean, I think, yeah, definitely.
I mean, the thing is, when you start making a lot of these measures, you have to be, if you're not a physician, you're not licensed to do it, you have to be very attentive, because you can get yourself in a lot of trouble. But pulmonologists do these things all the time, and I have no idea whether they would be expected to change with a breathing practice.
Right. Yeah. Something to explore.
I mean, do you deal with individuals who have muscle weakness and have difficulty ventilating because they have muscle weakness?
I would say yes. I think mechanically, maybe not necessarily, well, I guess as a result of muscle weakness. But I've had, some people who have a hard time even bringing the breath into their chest, or vice versa, even a low diaphragmatic breath, so I imagine there's some weakness, whether that's muscle or otherwise. So, that's definitely something I've worked with in the past.
Have you made measurements as people improved in their function?
I have kind of more like a range of motion type of measurement, with a tape, measuring tape around someone. I've definitely done that and seen a dramatic difference once we can kind of restore that breathing function into that part of the body. Yeah, I guess, I don't know. The question came from, obviously we have a couple hundred people, part of our community, doing a breath practice. And so how do we turn that into something quantifiable or create a study of sorts around that? I don't know. My head's just been kind of thinking about that. I don't know what that would be, but just, hey, we're breathing 12, 15 minutes a day. And, here's sort of what you can actually expect. Yes, we can say things like blood pressure, a slew of potential outcomes, but to actually have some data.
I would look at, I would Google pulmonary function tests.
Okay.
And see what might pop up. For example, something that might be easy and you might not have to worry about, a human review board is like expiratory flow. You can imagine blowing into something where there's a little ball, and the harder you blow, the higher the ball goes up. And that would just be a simple device that you measure how much force you can generate. And that is going to be a reflection on certain aspects of your musculature. I'm sure you could do the same thing with inspiratory flow. Other measurements start getting more complicated. What is the total lung capacity? What is the functional residual volume? Those become very complicated.
Right.
And here I would just suggest speaking to a pulmonologist, and you get a pulmonologist interested in it, interested in what you're doing, and I imagine that wouldn't be too difficult. They may be able to work with you to find things that can be done that are non-invasive. Measuring the chest gives you some indirect measure of the excursion of the movement, but there's a lot of things that are probably more direct, like forced expiratory flow, because really this is about movement of air. It's not about how you contract your muscles, but when you get the air out, you get the air in.
Right. Cool. Yeah, that's a great idea. I'll definitely, I mean, being in San Diego, we have UCSD here, which I would imagine there's probably some interesting minds there that would be keen to chat about it. So yeah, it's a great suggestion. Thank you.
Send me an email and I'll give you some names of people you can contact.
Cool. Thank you.
Because I know the head of pulmonary medicine there, and he has a lot of trainees and young guys and women, and they might be interested in, pursuing this.
Cool. That's great. Thank you so much.
That's interesting. So would this be into the categorization of like spirometry and capnometry? And just thinking, as Reese mentioned, there's 200 plus people and growing every week that are joining for these live morning breathing sessions. And so whether there's a pre-testing and then a post-testing at the end of the month to see, what type of metrics are shifting and how this is, because it can be quite subjective if there's a question of how
I
you
would
feel.
speak to these guys because they see patients all the time. They work with humans. So these guys are known. They're really sharp, and they're very engaging. So I think you could talk to them. I mean, if there's a simple, inexpensive device that everyone could have, that would be fine. Some of this equipment can get to be pretty exotic and expensive, but some of it may be relatively inexpensive, and it might be fun for you to do a experiment, to see before and after.
Yeah, absolutely. That's great. Thank you so much.
You've got me hungry to hear about episodic hypoxia.
Okay. The reason I mention it is Sam put me in touch with James Nestor, and I talked to him yesterday. We had a great conversation. Thank you, Sam. And I told him about episodic hypoxia, which he wasn't aware of. And so if I give you a gas mixture to breathe that is low in oxygen, so hypoxic gas mixture, let's say for 9 minutes, while you're breathing that hypoxic gas mixture, your ventilation will go up. And then if I give you room air again, it goes back down. Okay, pretty straightforward. If instead we did it 3 minutes hypoxia, 3 minutes normoxia, room air, 3, 3, 3, 3. So still 9 minutes hypoxia, but it's episodic. What happens is your ventilation goes up, down, up, down, up, down. And after the last episode, your ventilation keeps rising for the next hour to several hours. It stays elevated. So if you do it continuously, it goes up and it comes back down to baseline. If you do it episodically, it goes down and then it comes up and it stays up for hours. So this episodic hypoxia has been studied very intensely. And it seems that episodic hypoxia has positive benefits in cognition. So someone exposed to episodic hypoxia, if you then ask them to, give them a problem or whatnot, they seem to perform better. It seems to affect athletic performance. You have episodic hypoxia, and you go perform some athletic feat, you seem to be able to do it better. The main research center for this is now at the University of Florida, and the person there was Gordon Mitchell, who I'm happy to put you in touch with. He's great. And they've been studying this for a long, long time, and they found all sorts of things that are quite interesting about this. They're using it, for example, to try and deal with spinal cord rehabilitation. So there was a study done at Northwestern where an individual had a stroke and he had weakness with ankle extension. So they took this individual and put him in a chair where there was a little lever which he could extend his ankle. They can measure the force. Under control conditions, he was able to exert a modest amount of force. Then if they gave him episodic hypoxia, immediately afterwards, the force went way up. And this seems to be highly reproducible. So they actually have a study now where they're trying to look at spinal rehabilitation, that exposing people to episodic hypoxia can be beneficial in the long term in improving movement for spinal cord injury. And relative to your question, it may be contributing to rewiring of circuitry. I mean, we don't know. And they gave the Nobel Prize 2 or 3 years ago for the scientist who figured out what hypoxia does to cells. And when you have long-term hypoxia, chronic hypoxia, for example, you have heart failure, that's very bad. You get inflammation, you get all sorts of sequelae that are just really bad. And what they found out is that this hypoxia turns on sets of genes called HIF gene, hypoxia-induced factor genes. And those lead to all sorts of things that are, on the long term, not very good. It's also relevant for solid tumors, because at the center of solid tumors, they tend to be hypoxic. And so you get this sequelae going on that seems to make the cancers resistant to a lot of treatment. I think that it's this episodic hypoxia, okay, so why does episodic hypoxia work and not chronic hypoxia? Well, I don't think the nervous system evolved to punish you if you have episodic hypoxia. That's not the way a selective natural selection goes. I think these HIF genes are playing an important role that's good, but if you have chronic hypoxia, it turns out to be bad. What's the good part? The good part is you see an episodic hypoxia. That is, you have a cell becomes hypoxic transiently, and you turn on these genes, and these genes, when you turn them on transiently, seem to be acting in a way to say, what can we do to improve oxygen utilization in a cell when it's hypoxic? And what they seem to do is they seem to act on the mitochondria, which are the part of the cells that are important in the utilization of oxygen. And so this episodic hypoxia acts on the mitochondria to improve the mitochondrial performance in hypoxia. And that seems to have all sorts of benefits for cells. And moreover, Gordon Mitchell and his colleagues have shown that the episodic hypoxia causes the release of a variety of neurotrophic factors, including one called BDNF, which is brain-derived neurotrophic factor, which seems to be beneficial for a lot of things in the nervous system. I think that you all know about HIT, high intensity interval training. I think one of the reasons it works is you get this episodic hypoxia. It's not lasting for hours. You do a 20-minute HIT, and then it's over with. But what's happened is the cells become transiently hypoxic, and they restore, hypoxic restore, and then they want to improve. And so you turn on these genes, and these genes are tweaking the cells around. And so I think that's another way, depending on what the breathing practice you're using, if you become episodically hypoxic, like you hold your breath for a long time. Now, there's a difference between the episodic hypoxia they did experimentally and holding your breath. When you get exposed to a low oxygen mixture, you hyperventilate. So even though your cells are still exposed to low oxygen, your CO2 levels drop. So it's episodic hypoxia and episodic hypocapnia. You hold your breath, you can become transiently hypoxic, but you'll become hypercapnic. And it's very possible that the sequela of what a cell does will vary considerably, whether it's hypocapnic or hypercapnic, because that will affect the pH. And the pH has a profound effect on the cellular metabolism. So this is something you might think about as you look in your breathing practice to see what the transient effect might be on oxygen and CO2 in the blood, and whether or not you might consider episodic hypoxia playing a role independent of whatever the breathing movements themselves are doing in terms of signaling into the brain.
Is there a general like oxygen percentage that they're using when they're administering this?
You have to look at their papers. I think they're using like 8%.
Oh, wow. Okay.
But to be candid, Gordon and I have an experiment we were going to do until COVID hit. And that is that to see whether it affected athletic performance playing golf. So the idea is we're going to get a group of scratch golfers, and on one day, they'll have the same protocol on 2 days. Each day they will breathe from a gas mixture episodically, but on one day it'll be a hypoxic gas mixture, another day it'll be room air. And we'll measure the parameters of their golf game, how far they hit it, how straight they hit it, what their putting is and whatnot. And I told Gordon that if you want to monetize this, golfers would pay anything if you could sell them something that will give them the slightest advantage. But I think there's a reasonable chance you will see a measurable change in their performance. No, not guaranteed. Just of the effect. This is not transient. We're not asking them to do a breathing practice. This is just transient. But Gordon Mitchell is also doing experiments where they're doing this a bit every day and seeing what the long-term effects are. So I should put you, Sam, I should put you guys in touch with Gordon.
That's, yeah, amazing.
I'm
That would be
guessing,
great.
Gordon's a super guy. He's super nice. He's very articulate. And he's very enthusiastic. So you'll enjoy talking to him.
I'm guessing you're familiar with the research on hypercapnic exposure that people have done and the anxiety relieving effects that has had on patients.
Hyper or hypo?
Hyper, so
Hyper.
administering like 30% CO2.
Well, that
For-
usually produces
Yeah.
panic attacks in normal people. You're
But
saying,
Dr. Feinstein, who's been doing some research with that in his labs, Justin Feinstein.
right?
And I think James explored that, that he included in his book. It was really, they did an interesting study where there were two twins that had calcification or amygdalas that weren't operating as a normal human, and they admit they had never experienced subjectively any sense of fear or panic. And then after administering this 30% CO2, experienced the gasping and this sense of breathlessness.
That'll produce a panic attack. It's extremely unpleasant.
Yes, so James Nestor writes in his book about how it does induce a panic attack, but the panic attack is short-lived, and after that, it's thought because of the the lingering effects in elevating CO2 tolerance, that it medium term relieves chronic anxiety. So when you were saying these two different administrations of gas cocktails, I wondered, well, why not a third? Because I wonder if these people, after having their panic attack, would then proceed to putt with greater calm.
To be candid, I would not want to participate and do something that will cause people to have panic attacks. Panic attacks are extraordinarily unpleasant, and moreover, there are individuals who've never had a panic attack, but once they have a panic attack, it sensitizes them. They become so afraid of another panic attack that it actually can induce it. So I would stay as far away from that as possible, do it in. I would just stay away from it, because you've been. I mean, if you want to do it to yourself, be my guest, but I can tell you it's you couldn't do it.
That makes sense.
Yeah, I mean, if you ever speak to anyone who has panic disorder, it's extraordinarily. They describe it as extraordinarily unpleasant. And there are treatments for it, but when they're having panic attacks, it's just extraordinarily unpleasant.
Is there a way to induce that episodic hypoxia without a gas mixture?
I don't think so. Well, you can do that by holding your breath.
Right.
But then you're becoming hypercapnic.
Right. That, yeah.
Don't know. I don't know. Gordon would know this, whether anyone has done that experiment. And remember that when they do the experiments, they're giving the people hypoxic mixture for several minutes. Most people can't hold their breath for several minutes. So, maybe holding your breath for short periods of time will do it, but I'm not aware of any experimental data. But my lack of awareness should not mean that, does not mean it's not there. It's not something I've paid much attention to.
Interesting. I think then in terms of measurement for that, to do it without an external source of air would be that we'd need a capnometer to measure CO2 levels, and we could go into a very low and slow under breathing technique and then get the CO2 level up.
Yeah, the CO2 levels would rise, but that would not be the same thing as the experiments looking at episodic hypoxia. Now, that said, humans go through bouts of episodic hypoxia. For example, if you have obstructive sleep apnea, you go through periods where you have apnea and then you wake up and whatnot. And if you do that, instead of doing 3 or 5 episodes, you do it all night, then that turns out to be bad. It's a matter of dosage here. But in that case, during sleep, they're becoming hypoxic and hypercapnic, where the experimental protocol is hypoxic and hypocapnic. But these are things I would defer to Gordon to fill you in on, because he's like the world's expert on this.
Roger that. Well, we'd love to connect with him and would love to read the literature as well. Something that comes to mind, if I may, in regards to hypoxia and this episodic hypoxia exploration here, through some of the research I was looking at in connection to cold exposure as well, within the cold, and it also had connotation to hypoxia, that there's the activation of RBM3, the RNA binding motif 3, and its role in helping to rebuild the dendrites at the synaptic connection. I was curious for your take on that, if hypoxia does in fact have that activation and its potential driver for plasticity.
It's hard for me to focus on one thing when you have hypoxia, because it basically affects everything. So there's no doubt that it does affect this binding protein, but if you can't isolate it. So the answer is yes, it can affect it. It can affect plasticity. It can do all these things. But that's when you look at that in isolation. When you get exposed to hypoxia, it's not happening in isolation. And so I don't know enough about the experimental protocols to which it's controlled for, that is a singular effect that you can count on for hypoxia to induce synaptic plasticity. Because on the other hand, depending on how severe the hypoxia is, cellular metabolism gets disturbed, all sorts of things get released, and not all of them are beneficial. So I think biology is really complicated. I wish I could give you a better answer. I just, we don't know. And I personally don't like to latch on. I know in the scientific world, you find one thing, you latch onto it and you write it as hard as you can. But when you're trying to explain phenomena like breath practice, there are so many things that are being impacted that I think you have to be very circumspect in pushing it. Yeah, it can happen. But it doesn't change. You know, if you showed me that it was markedly different in different breath practices, and you associated the different breath practices with different changes in emotional cognitive function, then maybe you can begin to tie a story together. So these are all great questions, and this is what we spend the time in our lab trying to figure out. But they're pretty complicated, and we just we don't know. Sorry, Sam.
I'm curious what's next for Jack Feldman.
Well, we have this developing idea about how the rhythm of breathing gets generated. It's very novel, and we're trying to test it as much as we can. I think I may have talked to you guys about this before. About that, we talk about fireflies synchronizing. Did we talk about that last time? So the notion is you have these neurons which are firing randomly, but because of the way they're connected, they suddenly start marching together. And as they start marching together, the activity and the collective activity gets stronger and stronger and stronger. And then it suddenly takes off and you get a breath. And then it resets, and it's a low level of activity. And then they start synchronizing, and then it happens again. And it seems a little bit crazy because you wonder how you can have something extraordinarily reliable or when you have this random assembly every time. But the fact is that when you grind through the numbers, there are enough neurons that even if it's random, it's going to work every time. I mean, it's just almost counterintuitive if you actually do the numbers. The other thing about breathing that people forget is that it's extremely robust, but it's very labile. Your breathing changes can change like that. And it's very hard to build something that's both robust and highly labile. And we think that the lability is a natural consequence of this random assembly, that if you perturb it, it's very easy to push it off. So if you get up and are beginning to walk out, you increase your ventilation 3-4 fold. That happens like that. And we think that this is because the network is organized to be very labile, very stable if you leave it alone, but can change very quickly. And then, of course, where we are really in whole hog now and trying to understand the mechanisms by which breathing and breathing practice is modulating emotion and cognition. But how? So I liken it to say, like the idea that, okay, we do breathing practice, we change emotional state. That's a little bit like, I get in my car and I step on this pedal, the car goes faster. Okay. The phenomena is very clear, both for breathing and in my car stepping on the gas. But stepping on the gas in my car doesn't explain why the car goes forward faster. Okay, there's something going on, and of course we know what's going on. You feed more gas, or if you have an electric car, more current into the engine, and that the engine goes faster, and we understand a lot about how the engine works. Well, for breathing, we don't understand enough about how the engine works and how it influences and modulates these things that we call emotion and cognition. So that all into that, we're trying to set up a center for breathing and emotion at UCLA with a couple of people in psychiatry who use breathing practice to treat patients. That's another person you might want to talk to. You might want to talk to my partner in psychiatry who treats patients who have anxiety disorders and other emotional problems, and she treats them with breathing practice, and she is very effective. And she's very effective. She's really good. So Sam, if you drop me a note, I'll give you the contacts, and I'll CC these people to tell them that you'll get in contact with them.
Absolutely. Thank you. Will do, and look forward to that. Are you able to shed light onto some success stories there, techniques she's using? Is it a lower, slower type of breath? Are you aware of?
I would let Helen tell you.
Okay, roger that.
She seems to be, I shouldn't speak to her, but we were on a Zoom conference recently, and she was asked that question. And to paraphrase, and hopefully it's not, I'm not overstating things. She doesn't feel that the differences in the different breathing practices has much of a direct impact on the effect. That different practices can still have profound effects. But you will probably have specific questions, and she can give you the answers because she's much more knowledgeable about this than I am.
Okay, excellent. Well, we'll look forward to that as well then. So much to continue exploring, and we'd all like to respect your time. It's such a pleasure to be here with you. Time flies when you dive into the neural origins of breathing and all these topics. So I'd love to just cue for anybody who is hungry for a question. Are you okay with another question or two, Jack?
Yeah, I have to go in a few minutes. I should say, when you ask what the things we're up to, every couple of years I write a major scientific review about breathing. We wrote one in 2018, The Breathing Matters, which I think you guys read. So we're writing another one now. And we're trying to sort of explain all these things. And things have happened since 2018. And it's quite a challenge to take the very detailed scientific papers and put it in a format that even a general neuroscientist can understand. And so it's occupying a lot of our time, but we feel it's an important thing to do. Are there any more questions? I'm happy, Sam, if someone wants to email me directly, I'm happy to do this. I'm happy if there are enough questions that pile up to have another meeting. It's a delight to talk to you guys. And
I have a question,
sure.
if you don't mind. Yeah, I was just curious, we've been exploring, one of the basic elements of breath is that it's both an unconscious action that we can then take conscious control of. And that's a lot of what we're investigating. And I'm just curious from a neurological perspective, does a conscious breath look different than an unconscious breath? Like neurologically, what's happening? Is there something significant or interesting there to speak to?
That's a very interesting question. So we did the following experiment. We, through some molecular magic, we had the way to rapidly turn off the prebutsular complex in an awake rat. We did these experiments in rats. And we thought that when we stopped the prebutsular complex, the animal would volitionally breathe, which is what I think I would do if I noticed I wasn't breathing. We did the experiment. The animal stopped breathing. It apparently was aware that it wasn't breathing, but it generated no breathing movements, none whatsoever. So we would have thought that the volitional aspects of breathing would be able to directly control the diaphragm, for example, have a direct line to the diaphragm. Apparently, when you knock out the prebutsular complex, that's not the case. So our current thinking is that the volitional movements are routed through the prebutsular complex. Now, that said, the pattern of breathing is under your volitional control, and you can make it whatever you want. You can make it, if I showed a normal breath pattern, and was, for example, with airflow, and then measuring your actual airflow, you could pretty much follow that pattern. So yes, you can mimic it, but you can deviate however you want. But I don't think there's anything natural about breathing according to very close to the same pattern. Remember, what you're calling the involuntary breathing, I call it regulatory automatic. So because I don't like the voluntary, involuntary, it's regulatory, is highly tuned for efficiency because you're doing it 24/7, 365. And if you look at the energetics of breathing, you look at the amount of energy used to the various muscles involved in breathing. You look at the energy they expend at different tidal volumes, different changes in airflow, different frequencies. At rest, unless you're ill, you're right at the minimum. So there's a U-shaped function, for example, it's a function of frequency. So you're probably breathing 10 to 12 per minute. If you breathe faster or slower, you're going to burn more energy. This is wired into the system. When you do it volitionally, particularly transiently, it doesn't care anything about efficiency. I've often thought it would be a great way to get people to lose weight if you could teach them to breathe inefficiently and it wasn't uncomfortable. But when you start breathing inefficiently, it starts to be uncomfortable. Because then you could be blowing off calories and being oblivious to it. But right now you're at your minimum and you're not going to deviate far from it for the regulatory breathing. But when you do volitional breathing, it could go It's not restricted by, it's not bound by that. Does that answer the question?
Yeah, yeah, definitely gets to it. And I guess a second, like a follow-up would be, is there something that you learn from the neuroscience because this is such a unique circumstance where we have conscious control over something that's part of the regulatory system in the brainstem, that pathway from the cortex or the volitional brain to the brainstem, is that something that is studied, or is that like getting into, because this is like a bidirectional conscious way of engaging with your nervous system, which is pretty unique and new for me to be considering. And I'm just wondering if there's, what's in that, in terms of the science, being able to communicate between the brains.
But you're using your brain all the time. You're using your hands. I mean, you have enormous amount of ways that the brain is interacting with the body. We don't think the projection is monosynaptic, that is direct from the motor cortex to the diaphragm. We don't know particularly what the intermediate pathways are, but it does not appear to be direct. We've looked at that. And so I can't tell you what that pathway is. We actually have a grant proposal in to work with this brilliant neuroanatomist to try and map not just to the neurons that are projecting directly into prebuccear, but the neurons that are projecting to neurons that project into prebuccear. And then go back another level and see how long it takes us before we're in the motor cortex, in the set part of the motor cortex that's affecting the diaphragm. So it's a great question. We just haven't gotten to it yet.
So that's still emerging science, that part of it.
Yeah.
Thank you. Really.
My pleasure. You guys are paying for it, so you're entitled to know what's going on. It's our tax dollars, your tax dollars. My tax dollars too. Sam, I got to get going. Let's do this again sometime before too long. Don't worry that might appear to be too busy. There's always some way to carve out an hour. So if the situation arises, you want to talk some more, I'm happy to do it. You guys are going to shoot me some emails and I'll respond.
Excellent.
Okay.
Jack, thank you so much for your time. It's been a pleasure. Look forward for some follow-up information and onto the trail of some of these resources. Would love to have you on again, and really appreciate everyone's participation and questions, and really to acknowledge the work and the continuation of this work that you're doing. It's so encouraging and exciting. So
Well,
thank you.
And to the degree that you have outreach, spread the word. I mean, I think I mentioned it. I've been in my academic silo for so long, and I didn't fully realize the number of people who have some interest in this. And we're not trying to hide the information, but it's just finding a way to sort of get it out. And any ideas that you have, and certainly pass this information along. Okay, you guys take care, stay well and sane, stay healthy, and we'll see you soon. Bye now.
Thanks so much, Jack.
You too, Jack.